All right, let's dive into a truly remarkable technology, one that basically works like a detective at the molecular level. We're talking about Fourier transform infrared spectroscopy, or FTIR for short. It's an incredibly powerful way to identify unknown stuff just by reading its unique molecular fingerprint.
I mean, it sounds like something straight out of a sci-fi movie, right? You've got some mysterious powder or liquid, you zap it with a machine, and boom, in just a few moments you know exactly what it is. But this isn't fiction, it's real science, and we're going to break down exactly how it's possible.
So, here's our game plan. We'll start with the big idea of a molecular fingerprint. Then, we'll see why FTIR was such a game changer. After that, we're going to look under the hood at the machine's core, the interferometer. Then comes the fun part, learning to actually decode the results. And finally, we'll see where this incredible tech shows up all around us.
Okay, so the whole secret behind this technology comes down to one simple but really powerful idea. Every single molecule has a signature that is completely and totally its own, just like a human fingerprint. So think of it like this. When we shine infrared light, which is basically just a type of light we can't see, through a sample, the bonds holding the atoms together start to vibrate. They stretch, they bend, they kind of dance around, and as they do, they absorb very specific frequencies of that light. The pattern of which frequencies get absorbed creates this one-of-a-kind signature. That's the molecular fingerprint. Now, using infrared light to study molecules has actually been around for a long time, more than 70 years. But the FT part of FTIR, that stands for Fourier transform, that's what turned everything on its head. It took a slow, clunky lab method and made it into the super fast, powerful tool we have today. You see, the old way was, well, it was painfully slow. The machine had to scan through every single infrared frequency one by one by one. It could take ages. But FTIR, it does something amazing. It measures all the frequencies at the exact same time. It captures that entire fingerprint in a single shot. And capturing everything at once gives FTIR some huge advantages. First off, it's incredibly fast. We're talking scans that take seconds, not minutes. That's what scientists call the Felget advantage. It's also way more sensitive because much more of the light actually makes it through the system, which is the Jacquinot advantage. This means you get a much cleaner signal and can spot even tiny amounts of a substance. On top of that, the machine is pretty simple mechanically and uses a laser to constantly check its own accuracy, making it unbelievably precise. So how does it do that? How does it measure all those frequencies at once? To figure that out, we need to pop the hood and look at the real heart of the machine, a device called an interferometer. The whole process is actually a really elegant 5-step flow. You start with a source that shoots out a beam of infrared light. That beam goes into the interferometer, where it gets encoded. Then, the encoded beam passes through your sample. A detector on the other side measures what comes out, and finally, a computer does some quick math, the Fourier transform, to give us our answer. Now, this is that clever device I was talking about, the Michelson interferometer. It uses a special kind of mirror called a beam splitter to divide the infrared light into two separate beams. One beam bounces off a mirror that stays perfectly still. The other hits a mirror that's constantly moving back and forth. Because one beam travels a bit farther than the other, when they finally come back together, they interfere with each other, creating a brand new complex signal. And that complex signal is called an interferomogram. Now, you can't just read it directly. Think of it more like an encrypted message. But here's the key. Every single point in this messy-looking signal contains information about all the infrared frequencies at the exact same time. That's the secret to FTIR's speed. And here's what that raw signal actually looks like. Yeah, it's kind of a mess, right? But it's absolutely packed with information. See that huge spike right in the middle? That's called the center burst. It's the one special point in the mirror's travel where both light beams have traveled the exact same distance. So all the light waves are perfectly in sync, creating this massive peak. Okay, so we have this jumbled, coded signal. How in the world do we translate it into something we can understand? Well, this is where the magic happens. It's all thanks to a brilliant piece of mathematics called the Fourier transform, which became practical to use in machines back in the 1960s. And bam, this is the payoff. The computer runs the Fourier transform algorithm, and in a flash, that messy interferogram gets decoded into this beautiful, clean spectrum. This is our molecular fingerprint, showing us exactly which frequencies of light the sample absorbed. So we've got our fingerprint, but now for the most important part. How do we actually read it? How do we look at these peaks and valleys and turn them into a positive idea of what we're looking at? This is where the real detective work begins. Let's take a look at a real-world example. This right here is the spectrum for vanillin, the molecule that gives vanilla its amazing flavor and smell. Every single one of those sharp downward peaks isn't random. Each one is a clue, representing a specific type of bond inside the vanillin molecule, vibrating away as it absorbs a very particular frequency of light. This quote really nails it. The bonds that hold atoms together aren't like rigid sticks. They're much more like tiny springs. Different kinds of bonds, say a carbon to hydrogen bond versus a carbon to oxygen double bond, they all vibrate at different predictable frequencies. So they absorb different colors of infrared light. And that's what creates the peaks we see in the spectrum. And this table? This is basically the chemist's decoder ring. Through decades of research, scientists have mapped all this out. The peak's location is measured in something called wave numbers. So, for instance, if you see a big, broad dip over here between 3200 and 3600 wave numbers, that's the spectrum shouting, hey, I have an OH bond, probably from an alcohol. Spot a sharp, strong peak right around 1700? That's a dead giveaway for a carbon-oxygen double bond, which you'd find in a ketone. Now, while some peaks tell you about the general groups of atoms you have, the really crucial area for a positive ID is this part of the spectrum right here, from 1500 down to 500 wave numbers. It's called the fingerprint region for a reason. It's incredibly complex, representing all the intricate wiggles and bends of the molecule's entire structure. And that pattern is absolutely 100% unique to each molecule. There's just no faking. Okay, so we've dug into the science of how it works and how to read the results. Now, let's zoom back out and see all the incredible places this amazing tool is being put to work to solve real-world problems.
Take the pharmaceutical industry, for example. FTIR is an absolute workhorse. It's used for quality control on a massive scale, to instantly check if the right ingredients are in a pill, to make sure there are no dangerous contaminants, and to guarantee that every single batch of medicine is pure, consistent, and safe. But it doesn't stop there, not by a long shot. Art experts use it to analyze the pigments in old paintings to spot forgeries. Environmental scientists use it to sniff out pollutants in our air and water. It's even used to check the quality of the food we eat and the beverages we drink. And in what might be its most amazing application of all, FTIR isn't even confined to Earth anymore. NASA actually packed one of these instruments onto its Mars rovers. So right now, as we speak, this technology is on another planet, analyzing the chemical makeup of Martian rocks and its atmosphere, searching for clues about the past, and maybe even the potential for life. So from a lab bench here on Earth all the way to the surface of Mars, FTIR gives us this incredible power to read the hidden language of molecules. It really makes you wonder, if every single substance has its own unique story, its own fingerprint, what other incredible secrets are just out there waiting to be read?